All right, why don't we go ahead and get started. So the last time we were talking about the very basic details about T cell development, and I'm going to talk a little bit more today about T cell development, and then we're going to talk about tolerance and how that is attained inside of the thymus gland that we talked about last time. So just to remind you guys, my name is Craig Walsh, and this is my email address. And also, as it's listed in the syllabus, I have office hours on Wednesdays at 2 o'clock. So if you have any questions about lecture material the last couple of days, feel free to stop by. It's an open door policy, so just any questions, feel free to come by. All right, so today we're going to be talking, again, we're going to return for a brief moment and talk about allelic exclusion, and because I know there were some questions the last time, and it's kind of confusing, so we'll spend a little bit of time talking about that, and then we'll talk about positive selection and what we call lineage commitment, how a T cell decides whether it's going to become a CD4 helper T cell or a CD8 cytotoxic cell, and that's all taking place based on the antigen and the MHC that peptide antigen is presented by in the thymus. We'll talk about just briefly about the pre-T cell receptor, spend time talking about negative selection and this concept of an affinity model for selection. Basically this idea that what's really driving the selection, both positive and negative selection of T cells, is the affinity between the T cell receptor and its peptide presented by MHC. Okay. So the last time we talked about this concept of allelic exclusion. You guys have heard about this before in the context of B cells, and I really wanted to spend a little bit of time going over this just so it's clear. So one of the things that I showed you guys the last time is that T cells have the ability to become either gamma delta cells or they can become alpha beta cells. And these have different roles in the immune system. These gamma delta cells are sort of innate-like. They have a very limited number of receptors that are available. They're kind of like the lambda kappa distinction in terms of the light chain in B cells. Similar thing is going on with T cells. So the gamma delta cells don't recognize typical peptide antigens like we think. And we're going to return to this later when we talk about immune responses to infection. On the other hand, these alpha beta T cells, they're the kind of T cells that we normally think of in terms of being restricted to MHC. They bind to peptides that are presented by MHC, and they do all the job of, for example, acting as helper cells. They can, if they're CD8 cells, they can go around and kill cells that are, say, virally infected, or they're tumor cells and what have you. So those are the distinctions between those two different subsets. But one of the key things that's, I think, a little bit confusing about this process, in terms of discrimination as to whether the cell is going to become a gamma delta cell or an alpha beta cell is just, what's happening in these cells. So as I mentioned before, as these cells in their early double negative stage, they have this upregulation of the recombination machinery in the cell. So that you get upregulation of RAG1 and RAG2, and so there's rearrangements that are taking place on these three loci: the gamma locus, the delta locus, and also the beta locus. And at this point, you don't have rearrangements taking place at the alpha locus just yet. And so at this point, it's essentially a race. Okay, so if you have productive rearrangements that take place in the gamma chain, and you have rearrangements that take place in the delta gene that give rise to productive rearrangements, meaning that you've got appropriate VDJ or VJ rearrangement, the result is that those molecules can then be presented on the surface. These guys meet up in the ER, make their way out through the secretory pathway to the surface of the cell, and then that sends a signal into the cell that says, shut off recombination, we don't need to do anymore. We've got a gamma delta cell here. Alternatively, the cell is also trying to rearrange at the beta locus. And so if you get a productive rearrangement, VDJ rearrangement at the beta locus, then the consequence of that is that you get this pre-T alpha receptor. Okay, so you have this pre-T alpha protein that's essentially what we would call a surrogate T cell receptor that kind of takes place of a rearranged alpha chain. And so the result is that pre-T alpha interacts, it sets up a heterodimer in the ER with the productively rearranged beta chain. That then makes its way out to the cell surface and then sends a signal into the cell to shut off any additional rearrangement. So the RAG genes get shut off, there's no more rearrangement that takes place, and that essentially allows this cell to go down this alpha beta lineage. On the other hand, in order to get a fully mature alpha beta T cell, you need to have rearrangements that take place in the alpha chain. Okay, so a couple things happen here. So one of the things that happens is upon receiving this signal through the beta chain paired with this pre-T alpha, as I said, that shuts off rearrangement, any further rearrangement, so the cell is going to go down that pathway. It protects the cell from apoptosis. So if they don't get that signal, and they don't have a productively rearranged gamma chain paired with a productively rearranged delta chain, the cell dies. And we call that process beta selection. So we talked a little bit about that the last time. It's basically, if you've taken cell biology, you've probably heard about growth factor receptors. And if cells don't get these kind of growth factor receptor signals, they will die off. Okay, so it's just an important control mechanism that our cells have to make sure that they're in the right place at the right time. And the similar thing is happening in these T cells. So if they don't have that signal coming through a productively rearranged beta chain or have a gamma delta chain on the receptor on the surface, the result is that they die off. In addition to that, they start to proliferate, meaning that they undergo cell cycle, cell division, and you get 8 to 10 rounds of division at that time, upon which they then start to upregulate the CD4 and CD8 receptors on the surface. And we call those cells, that's Wednesday. Double positive, good. All right, so those are the double positives because they have 4 and 8 on the surface. And those guys, as we're going to see, are subject to this process of selection. Now, the important point of this is that they need to be able to get a productive alpha chain rearrangement. And they essentially have 2 shots at it on each locus, right? So you have a paternal locus and you have a maternal locus. And because of the way that VJ rearrangement occurs, that actually turns out to be 2 possible ways in which or 2 events that can take place on each the maternal allele and the paternal allele. So the result is that they essentially have 4 chances to do this. Okay, so the consequence of that is that if they finally get a productive rearrangement, that'll protect the cell from cell death as well, and we'll talk about that in a minute. Alternatively, these gamma delta rearrangements can still take place, right? So after they've done this proliferation following this beta selection stage, and they upregulate CD4 and CD8, they upregulate the rearrangement genes, RAG1 and RAG2. And so in addition to being able to rearrange the alpha chain, they can also, in theory, upregulate rearrangements on gamma and delta. And so if they can productively rearrange gamma and delta, the result is that at this stage, you develop a gamma delta cell. And it can then go out into the periphery and become part of the gamma delta repertoire. On the other hand, if you have a productive rearrangement of the alpha chain, the result is that the cell can then turn into an alpha beta T cell. As we'll see, it's subject to selection, both negative and positive selection, but if it then gets beyond that, then it becomes part of the peripheral alpha beta T cell repertoire. So when we think about T cell selection, there's an important point about this, and that is that when we think about the total possible number of T cell receptors that one can produce, it's in the billions, right? Because of the diversity that's generated, as you guys have learned about with respect to recombination that takes place on the T cell receptor gene loci, that recombination based on junctional diversity and so forth gives rise to this massive number of potential T cells that can be generated. And as you guys have learned, on these T cells, they have these what are called CDR1, CDR2, and CDR3 loops. These are the parts of the molecule that actually interact with MHC and peptide. And the CDR3 loop, and we'll talk about this more later, but this CDR3 loop is the part that's generated as a result of these junctions that occur between VD and J on the beta locus and VJ on the alpha locus. And so the result of that is that that's where the specificity for peptide is going to occur. But we also know that there are these CDR1 and CDR2, these complementarity determining regions 1 and 2 that exist in these V segments. And one of the key points of this is that in any one individual, evolution has developed all of these different T cell receptors, and there's been a significant amount of mutation and evolution that's taken place in CDR1 and CDR2 that allows those T cells to be able to recognize self-MHC. Now, why does a T cell need to recognize self-MHC? This is different from, so we call this self-restriction. This is different from self-tolerance, and we'll get into the distinction of that in a minute. But why would a T cell receptor need to be able to bind to MHC? Yeah? Right, in order for the T cell receptor to be able to recognize peptide, it also needs to be able to recognize the MHC itself. So the MHC, we can think of it as sort of a hot dog in a bun, right? The hot dog is the peptide that's strung through the middle of this thing, and the bun are these alpha helices on the top of that MHC molecule. All three of those, the two alpha helices, part of the MHC, as well as that little peptide that's strung between the two pieces of the bun, all of those have to be recognized by the T cell receptor. So it's recognizing, all of those components. And so instead of thinking about the interaction between a T cell receptor and MHC peptide as affinity, we actually call it the avidity. So has anybody ever heard the word avidity before? So when we say avidity, what do we mean as opposed to affinity? So it's the combination of multiple binding sites. It's the affinities, the additive affinities of all of those we would call the avidity. So there's contributions in terms of that, the amount of binding of that T cell receptor to peptide MHC that's being contributed not just by the peptide but also by the MHC itself. And so the result is that evolution has ensured that we have T cell receptors with the appropriate sequences in these CDR1 and CDR2 regions that allow these T cell receptors to be able to bind to MHC. But the important point of this is that the T cell receptor loci are distinct from the MHC loci. And as you guys have learned in previous lectures, there's limited repertoire of MHC in any particular individual. So all of us have at most maybe about 12 different class 1 and class 2 genes that give rise to unique MHC on the surface. And so a significant number of T cells that have, or T cell receptors I should say, in any one individual are not going to be very effective. And it turns out that there's a significant amount of T cells that have gone through what we would consider to be productive rearrangements of beta. They present an alpha chain on the surface, but because of the fact that the T cells are not going to be able to bind to self-MHC, those cells are going to be useless in the periphery, and they end up dying off. They're not getting these signals through their T cell receptor that allow those cells to survive. And so we call that process death by neglect. So basically the idea is that if a T cell receptor is produced that can't bind to our self-MHC, it has no affinity for self-MHC, then it's obviously going to be useless in the periphery. It's not going to be able to detect, say, a viral peptide or something like that. And so the result is it makes sense to get rid of those, and those cells then end up dying apoptotically inside of the thymic cortex. All right, so I'll take a break here and see if anybody has any questions about this. It's a little bit complicated. Yeah?
Yeah, so Dr. Fruin was talking about rejection of organs because those organs would express MHC molecules that aren't produced in the patients.
Mm-hmm.
So wouldn't these TCR receptors that aren't self-MHC be beneficial for those patients? Does that make sense?
So you're talking about alloreactivity. It's the idea that the person next to you decides that they're going to donate their kidney to you because your kidney's failing or something. You're going to reject that kidney. We have a whole lecture on this. We'll talk about more later, but it's an important point. The T cell receptor is evolved to be able to bind to MHC and not necessarily your own MHC. So there will be some T cell receptors in your T cell repertoire that are capable of binding to what we would call an allogeneic MHC from somebody else. But it's not beneficial. In that case, it's actually going to cause rejection. And so if you got a transplant from somebody else that didn't have exactly the same MHC molecules, then you will have T cells in your repertoire that are capable of recognizing that and then setting up a rejection response. Any other questions? Yeah?
Figure 7.7, where can you see the checkpoints? What would it be?
When we talk about checkpoints, we'll come back more to this later, but the checkpoints that we'll talk about are basically, for example, here, where the beta chain is first being recognized, and then later during this process of positive selection, where the cell needs to discriminate whether or not it's getting a signal. Okay, any other questions? All right. So again, I take my teacher's prerogative and we'll see a movie. And this particular movie just kind of describes the process of selection, and we'll get into more details in today's lecture. But the important point being that, you know, all of these selective events, whether it's to produce a T cell that is capable of binding to MHC, and whether that cell is tolerant of self, whether it's, you know, going to bind to our own tissues and cause problems, or be self-tolerant, that is all dependent on signals that are solely coming through the T cell receptor. The avidity of the T cell receptor interaction with MHC and peptide is solely responsible for all of these
T cell development
events.
takes place within the thymus, a specialized organ where T cell progenitors mature and differentiate. The thymus is divided into several different anatomical locations,
Oops.
to another. T cell progenitors enter the thymus through high anatomical
Sorry about that.
locations, and as thymocytes mature, they pass from one environment to another. T cell progenitors enter the thymus through high endothelial venules and migrate to the subcapsular region, where these thymocytes start the process of maturation by rearrangement of their T cell receptor genes. As maturation proceeds and the cells begin to express a specific receptor, they move further into the thymic cortex. Interactions between the T cell receptor and MHC peptide complexes displayed by the thymic cortical epithelial cells play an important part in the fate of the thymocyte. At this point, the developing thymocytes express both CD4 and CD8, as well as an alpha-beta T cell receptor. A cell that has a receptor able to recognize an MHC class I molecule receives both a survival signal and a maturation signal. Eventually, the cell stops expressing CD4 and maintains expression of CD8. Thymocytes that obtain a survival signal are said to undergo positive selection. A cell that has a receptor able to recognize an MHC class II molecule also receives a survival signal and a different maturation signal. Eventually, this cell stops expressing CD8 and maintains expression of CD4. Thymocytes whose receptors are unable to recognize either MHC class I or MHC class II molecules fail to receive any survival signals and die by programmed cell death or apoptosis. Thymocytes that are able to recognize MHC class I or MHC class II peptide complexes too avidly receive a strong signal that drives them into cell death. In this way, thymocytes capable of responding to self-peptide antigens are eliminated in a process known as negative selection or central tolerance. The surviving T cells migrate from the thymic cortex to the medulla. At this stage, they remain capable of recognizing self-peptide antigens expressed on other cell types, such as dendritic cells or thymic macrophages, and receive a sufficient signal to cause them to undergo programmed cell death. The remaining thymocytes, now mature, naive CD4 and CD8 T cells, pass out of the thymus, either returning to the bloodstream directly, passing into venules, or via the lymphatic system.
Okay, so there's an important point that was brought up in this video, which is that, you know, as I mentioned before, the T cell receptor interaction with MHC peptide is really what's driving both positive and negative selection. And we can kind of think of it as sort of a Goldilocks model, right? It's not too avid, you know, not too hot, not too cold, and that's really what's involved. So if it's not avid enough, if you don't have enough interaction with the T cell receptor, the cell's going to die off. And so that's part of positive selection that we'll talk about. The second part of positive selection is whether the cell is going to become a CD4 or CD8 cell. So we'll talk about that. And then the other part of it is negative selection. So if the interaction between the T cell receptor and peptide MHC is too strong, then the result is that sends a signal into the T cell that says, you're autoreactive, let's kill you off. And as we'll hear, that's that's sort of an iterative process. So it can happen inside of the cortex, and it can also happen in the medulla, and that simply depends on where those antigens are presented. All right, so let's first talk about positive selection. So as I mentioned, there are really two things going on here, one of which is to simply recognize whether the T cell receptor is capable of binding to self-MHC. And so an example of that shown here for an alpha beta expressing T cell. So it's productively rearranged the alpha chain, it's productively rearranged the beta chain. That's on the surface of this cell. And within the cortex, remember we were talking the last time about these stroma. You know, that's just kind of the overall bag that holds the thymus together. Well, there are these cells, these epithelial cells that are part of that stroma that can present MHC on the surface. And so the result of that is that you know, the T cells that are coming through have these TCRs that are recognizing self-MHC. That sends a survival signal into the cell that allows that cell to survive. And keep in mind that this cell obviously is recognizing not simply, you know, this MHC that's presented on these cortical epithelial cells, but on top of that, it's also recognizing any self-peptides that are there, because there's nothing special about these MHC molecules. They are designed to pick up peptide antigen, and they're presenting it. And of course, that's going to be self-peptide. So there's going to be a lot of self-peptide expression that's present on the surface of that MHC molecule. And the point of this is that, you know, you want to have this sort of moderate binding that allows the cell to survive. And so the point of this is essentially it's acting, like I said before, sort of like a growth factor receptor. It says, you know, hey, we're binding to MHC, which is what we're supposed to do, and we're going to survive. And if we don't bind to MHC, then we're useless, and we're just going to take ourselves out. And that's exactly what happens. These guys, if they don't bind to MHC, they're then going to go on, commit suicide through apoptosis, and you know, this process is extremely inefficient. So it's roughly about 2% of the total T cells that are coming through that have productive rearrangements that then go on and die off because they are not binding to MHC. Yeah?
Just a simplification, since there's no CD4 or CD8.
I'm sorry, I couldn't.
Is it just a simple cut through since there's no CD4 or CD8? I thought it was part of the positive selection.
Yeah, it's not shown here, but this is happening on these double positive cells. So they do have both CD4 and CD8. And the interaction between the T cell receptor is going to be driving, as we'll see in the next slide, is going to be driving the discrimination as to whether that cell is going to ultimately become a CD4 or CD8 cell.
CD4 or CD8.
They'll have both. They'll have both. Any other question? Okay. So that's the first thing. We want to make sure that the T cell is capable of binding to MHC in the first place. And so the second part of this is what we would call lineage commitment, this idea that a T cell needs to decide whether it's capable of binding MHC class 1 or MHC class 2 and become either a CD8 cytotoxic T cell or a CD4 positive helper T cell respectively. And so that's shown here in figure 717. And so here you can see this interaction. Again, it's with the same cells, these thymic epithelial cells, on the surface of which we have presentation of both MHC class 1 and MHC class 2. And let's say a T cell comes along and its T cell receptor binds with reasonable avidity to peptide presented by MHC class 1. So the result of that then is this cell, through that interaction, is going to then downregulate CD4, maintain CD8 expression, and then it's going to go along and become committed to a CD8 cytotoxic T cell. On the other hand, if the peptide MHC complex at that particular T cell receptor, again, it's undergone all the rearrangements and so forth, that particular T cell receptor recognizes peptide presented by MHC class 2, then it doesn't make sense for the cell to go down the CD8 lineage. Instead, it's going to become a CD4 cell, downregulate CD8 on its surface, and then turn into a helper T cell, and then exit the thymus and become part of the helper T cell repertoire. Okay, so does anybody have any question about that? Yeah.
Is this for there's no commitment? Is that what leads to DeGeorge syndrome with these thymic epithelial cells? They're not.
Oh, DeGeorge syndrome has more to do with just not having those epithelial cells in the first place. Okay. Any other questions? so how does this work? I mean, what's the signal that tells the cell that it's bound class 1 or class 2? Well, there's basically two different models that one can put out there to explain how this might work. And you know, this is from several years ago, but it's important to note how this is kind of formulated and how we know what we know about this. So one model is the stochastic model, which is basically that as the cells, you know, these are double positive cells, they have both CD4 and CD8 on the surface, and that they have essentially a random process where they downregulate randomly either CD4 or CD8 during the process of interaction with MHC and peptide. And so the result is that you have a certain fraction of cells that, you know, considerable fraction of cells that have downregulated the wrong receptor, and they're just going to die off by this process of apoptosis that I talked about before, where, you know, if they don't have this strong interaction, they will die by benign neglect. Okay, so that's one possibility. Basically just a random process, downregulate either CD4 or CD8, and if you make the right choice, you become one of those two. If you make the wrong choice, then you die off by benign neglect. The second model is what we call the instructive model, and that is basically that there's signals inside of these cells that are unique. So CD4, for example, may send a unique signal to that T cell that says, hey, we're binding to class 2, we need to turn into a helper cell. So how does that work? Well, it turns out that if you look at MHC class 1 and MHC class 2, they have these invariant portions. You know, we think about all the polymorphism that exists inside of our different MHC molecules, but there are regions that we can think of as sort of like the constant region where they don't change. And that particular region allows the T cell receptor to basically determine, or the T cell, I should say, to be able to determine whether it's binding to class 1 or class 2. And how does that happen? Well, class 2 is bound in this invariant portion by CD4. So there's a portion of class 2 that allows it to bind to CD4. And on CD8, there's a similar complex that's formed with MHC class 1. And so the result is that in this instructive model, there are distinct signals that the cell is getting. So not only is it getting that signal through the T cell receptor binding to MHC peptide itself, but you're also getting a signal that's associated either with CD4 that tells it it's binding to class 2, or with CD8 that tells it it's binding to MHC class 1. And so we understand a lot about the signaling that goes on with that, but we're not going to talk about that in this class. Okay. But the important point is that we now know that it's this instructive model that's correct. So simply by binding the T cell, CD8 or CD4 are going to send in signals into that cell that says whether they're going to then turn into a helper cell or a cytotoxic cell. Any questions about that? Okay. All right, so then we're going to move on to the process of negative selection, which is basically, you know, this part of the Goldilocks model where you have cells that are, you know, they're not binding too much, they're not binding too little, they need to be just right. And in this case, we want to make sure that we don't have cells that are overtly autoreactive. We want to make sure that they don't have a really, really strong avidity interaction with self-peptide and MHC, because otherwise that would then be a problem, right? You'd get these cells out in the periphery, which could then cause an autoimmune response. And so what happens with this is here you have an antigen-presenting cell. In this case, it's a dendritic cell, although we'll talk about different cells that are capable of presenting self-peptides in the thymus. And so the result of this is that T cell comes along, and if it has this moderate binding, meaning that the T cell receptor binds to peptide and MHC with sort of an intermediate avidity, the result is that that sends a signal into the cell that's pro-survival. The cell survives and then goes on and continues along in its differentiation. On the other hand, if that interaction is very tight, so you have the T cell receptor is not simply interacting very strongly with the MHC, but it's also interacting with self-peptide, then the result is that that sends a signal inside of the cell that causes it to commit suicide. And so the point of this, again, is that not only are we trying to get rid of cells that are not reactive at all, but we also want to get these autoreactive cells out of the system, because if we don't get rid of those guys, then they'll respond to self-tissue and then cause an autoimmune disease. All right. So at this point, we've gone through positive selection, and we've also gone through this process of negative selection. And essentially now what we have are cells that have survived both of those guys upregulate the T cell receptor. They've decided whether they're going to become a CD4 cell by downregulating CD8 or a CD8 cell by downregulating CD4, and they go into the medulla of the thymus. And so they're going to then continue along their maturation. They're going to additionally interact with antigen-presenting cells there that may induce additional deletion. So there's multiple points at which this process of negative selection can take place. Now, how do we know that this is all true? How do we know that, yeah, question?
Does it matter which APC cell is involved in negative selection versus positive selection?
So the question is, does it matter what antigen-presenting cell is involved in terms of negative selection and positive selection? And the answer to that is, it depends. So if there is presentation of strong peptide antigen, autoreactive or self-peptides on these cortical epithelial cells. I should say, if there's strong recognition of self-peptides in the cortex by these cortical epithelial cells, that will induce negative selection there. There are unique subsets of cells that we'll talk about in a minute that are present in the cortical medullary junction, the junction between those two areas of the thymus that are also involved in negative selection. And then there are medullary epithelial cells that also can promote this process of selection. Okay, so the cells that are doing this, both positive and negative selection, when we think of positive selection, we're talking about the cortical epithelial cells. Negative selection is really driven by multiple antigen-presenting cells. Any other questions? All right, so how do we know that this all happens? I mean, you know, it's a nice model, but how do you really show that this is the case? Well, one way that you can show this is by generation of something called a transgenic mouse. Has anybody ever heard of a transgenic mouse before? You guys probably, have you heard of knockout mice? So a knockout mouse that's a situation where you take there's a particular gene that you want to modify somehow. You want to, disrupt it. And so you then use homologous recombination in embryonic stem cells and then make that mutation, take those ES cells, put those into an early embryo, and then you get a mouse that's chimeric. You take the mouse that's chimeric, hope that some of those alleles get into the germline, and then you can breed them, and then eventually have a mouse that breeds true for that particular mutation. Transgenic mouse is a little bit different, and in fact actually predates the generation of, or the development of these knockout mice. Basically what it is you take a gene segment that you want to express inside of the mouse somewhere, and you introduce that into the male pronucleus of sperm. That is then used to fertilize what's called a pseudopregnant female, and then you generate mice that have that gene sequence that has become essentially randomly incorporated somewhere in the genome of this mouse. It's now present in that mouse. And if you developed it properly, oftentimes you'll do this with a tissue-specific promoter, where you get expression of this in specific tissues in this mouse. And so it turns out that if you clone the T cell receptor genes, in this case we have a T cell receptor comprised of an alpha chain and a beta chain, and you clone those two sequences and then you introduce those using this transgenic technology, you end up with a mouse that has essentially all of its T cells recognizing the particular antigen that T cell receptor, that alpha beta chain combination is capable of recognizing. Okay, so that's interesting in its own right, because it means that you've kind of gotten around all of this issue of allelic exclusion and so forth. You're not going to get any further recombination events taking place because the signals are there to shut off any further rearrangements for the most part. So in this case, scientists had identified something called the male HY antigen. So this is a particular Y-linked protein that's present in males, and it turns out that this is what we would call a minor histocompatibility antigen. So when we talk about histocompatibility, we were just talking a little bit about transplantation. Usually we think of the MHC, the major histocompatibility, which is giving rise to peptide presentation and so forth. But these minor antigens have been known about for a long time. One of these is this male-specific HY antigen. So this T cell receptor that's been then introduced into a transgenic mouse is specific for this HY antigen. And so the result is that now all the T cells that come out of this mouse are going to be specific for male HY in the context of this particular MHC allele. All right, so what happens then? So if we look in a male mouse, what you see, and again, we're looking back, I showed you guys this FACS plot, which, immunologists like to look at FACS plots, and this is just an example of that. And remember, we were talking about the double negatives, so we've stained here for CD8 on the x-axis and CD4 on the y-axis, and you have this population of what are called double positive cells. Those are the guys that are going to be subject to positive and negative selection. You have these double negatives, and those are the cells where you've got these early events taking place, including rearrangement of the T cell receptor beta chain. And then you have fully matured single positive CD4 cells and single positive CD8 cells that can then continue maturation, go out into the periphery, and become part of that peripheral T cell pool. So one of the things you might note here is that there's a difference in the number of these double positive cells in a female versus a male. So why do you think that is? Yeah?
All the double positive cells are self-reactive, therefore everything else.
Exactly. So these cells here, these guys are going to be both positively and negatively selected. And here in a male mouse, these male mice have this HY antigen, this minor histocompatibility antigen, that's going to drive significant negative selection because every single T cell, every single double positive thymocyte is reactive against that. And so that reactivity is going to then drive their negative selection. So that's why you get, and actually if you look at these mice, their thymus is tiny. You probably wouldn't want to use them for making sweetbreads. So tiny little thymus, very, very few of these double positive cells, and that's all being driven by massive amounts of negative selection. And furthermore, if you take a slice through this thymus and then you stain it, what you find is that most of these double positive cells are apoptotic. They're actually undergoing apoptosis that's being driven by this really strong signal that's coming through the T cell receptor. Now that's in contrast in females. And here, what do you see? You see, first of all, these guys have a large thymus, which is not apparent in this, but they have a big thymus, essentially a normal sized thymus. And you see that there's a lot of these double positive cells. So the double positives don't seem to be deleting. They're not apoptotic, so they're pretty happy. But on top of that, what you also see is that the fraction of, first of all, there's a lot fewer of these double negative cells in the females than there are in the males. And you see a lot more, especially these CD8 positive cells. And it turns out that this particular T cell receptor recognizes this male HY antigen in the context of MHC class 1, so it makes sense you would drive a lot of positive selection of these CD8 cells. All right, and we know, and this is just one example, there's a lot of examples, but we know that the signal itself, the strength of that signal is controlling what's going to happen to these double positive T cells, because here, in a mouse that has another genetic manipulation, they carry a transgene that blocks T cell receptor signaling. So they actually block the downstream pathways. By blocking those pathways, it essentially raises the threshold for T cell activation, both for positive selection and negative selection. And we know that because we get a lot more of these double positive cells. And on top of that, we also reduce the amount of these CD8 single positive cells that have been undergoing positive selection. So again, to stress the point, all of this is being driven simply by the strength of the T cell receptor binding to peptide and MHC in the mouse within the thymus. Any questions? All right. So one of the questions that we had was, what's the source of, what are the antigen presenting cells that are driving positive and negative selection? And in particular, with respect to, I mentioned that the cortical epithelial cells are really important for the positive selection because they're presenting self-MHC, and that's really what's driving that positive selection. But then, what about the negative selection? So there's a really important point here, which is that, if we want our immune system to be self-tolerant, we need to make sure that it's self-tolerant not simply to antigens that are inside the thymus, which are, of course, going to be most of the antigens, right, because they're housekeeping proteins like cytochrome C and so forth that are going to be present in all cells. But you guys know that each cell in our body has a specific subset of proteins that it needs to express that differentiates it from other cell types, right? So you need to have, you know, for example, your big toe antigens somehow need to be presented in the thymus. And if you don't, what's going to happen? Well, you're going to walk around with your big toe falling off because your immune system is going to respond to that. Once it gets out of the thymus, those T cells will recognize that and then attack that. So that's obviously a problem. So how does the immune system deal with all these different antigens that it needs to see? Well, one of the things that happens is that there are resident cells in the thymus. When we talk about resident cells in the thymus, what I mean are cells that are derived from the stroma. So we have, as I mentioned, cortical epithelial cells. And those cortical epithelial cells will drive negative selection early on as the cells are still undergoing the differentiation in the cortex. We also have cells that are bone marrow derived, and examples of those are macrophages and dendritic cells that are present in the medulla and this area called the cortico-medullary junction, which is basically this interface between the cortex and the medulla. And those cells are coming in through the blood. So they in a sense, because they're coming in through the blood, they're coming in through the lymphatic system, they can pick up peripheral antigens, and then they can present those cells, or those antigens, to these double positive cells as they're making their way into the cortex. And then third, we have a specialized subset of these epithelial cells called medullary epithelial cells. And what they do, which is unique here, is they have a transcription factor called AIR. So, you know, if you think about it, as we age, there are certain antigens that we don't present when we're babies. For example, you know, those associated with puberty and so forth. That you know, as we get older, we start to express a lot of those, but those are not going to be present when we're still in the fetus, when we're first generating our first repertoire of T cells. So that means, and because I mentioned earlier that T cells are fairly long-lived, that means that in a sense, you know, when you're still in utero, you're generating all these T cells that could potentially be autoreactive. So how does the immune system deal with that? So these cells express this AIR protein, which is essentially a transcription factor, and it leads to expression within these medullary epithelial cells of essentially all of these peripheral antigens. So antigens that may not normally be expressed in the thymus can now be expressed as a result of the action of this particular transcription factor. And so now the immune system is essentially deleting T cells for antigens that are not expressed anywhere else in the body, but at some point will be. Okay, so that's how we eliminate these T cells that are autoreactive, even against antigens that are not expressed anywhere within the animal. Any questions about that? All right. So we've talked a little bit before about how these different events are taking place within unique areas of the thymus. And I thought I'd go over that again. And the first thing I want to talk about are the events that take place in the cortex. So as we've heard, most of the development that's actually taking place, the differentiation, is happening within this double negative stage. And that occurs within this cortical region, which is densely staining. So there's a lot of these thymocytes that are present there. That's all taking place in the cortex. And these are mostly in this double negative stage where they need to rearrange the beta chain and then ultimately rearrange the alpha chain. That's all taking place here. And that's in particular taking place at the outside of the cortex in this area that we call the subcapsular region. So this gray thing right here is the capsule, which is essentially stromal cells. And then all of this early differentiation is taking place towards the outside. So they've moved into the thymus, make their way to this area, and they're basically following a chemotactic gradient of factors called chemokines. And we'll talk a lot more in this course about chemokines and how they are really important for getting T cells and B cells and so forth to the right place in the body. But they play a really important role in the trafficking of these early T cells. So they then move into the center of the cortex during this process of positive selection, where they then can interact with these cortical epithelial cells, which then allow them to, number one, determine whether they're going to become a CD4 or CD8 cell, and then also whether they can bind, the T cell receptor can bind to self-MHC. Then the next step is this process of negative selection, which can happen within the cortex here. So if the cells are overtly autoreactive and you get a very strong signal through the T cell receptor, that's going to then induce apoptosis. Now, in the medulla, this sort of sparsely staining part of the thymus, it's at this point where the cells have already decided whether they're CD4 or CD8, so they're in the process of downregulating the wrong co-receptor. They make their way into the medulla, and it's here that they're going to essentially hang out for another 2-3 days before they make their way out into the periphery. And it's in this context where we have potentially additional negative selection events. And in this case here, it's either cells that exist on this cortico-medullary junction as they're traversing this, or cells that are present within the medulla itself that can then present these self-antigens that can drive negative selection of cells that are overtly reactive. And then finally, they make their way to these venules and then can then leave the thymus, go out into the periphery, and do their job, that they need to do. So there's one last point that I want to bring up, and that is this, what I've talked about so far when we're talking about selection, and in particular with respect to negative selection, is this idea that you have essentially what we would call passive tolerance, this idea that by deleting these cells that are overtly autoreactive, you prevent autoimmunity. But we know that that's not true, and part of the reason for that is that there's a subset of cells that are called regulatory T cells, and these are essentially a special subset of T cells that when they recognize self-peptide, they become activated and they suppress the responses of other cells, of other CD4 and CD8 T cells. So why would you want to have these guys? Well, it turns out that the process of T cell activation is clearly driven by the T cell receptor interaction with MHC peptide. But there are other additional factors we'll talk about. An example of this is co-stimulation, the cytokine environment, and so forth, that if a cell that was just kind of weakly autoreactive doesn't get deleted in the thymus, it may make its way out to the periphery, and under certain circumstances, like a viral infection or something, may become activated. So you may have in fact, all of us in this room have autoreactive T cells in our body, and yet I would say most of us probably don't have some sort of an autoimmune disease. And the reason for that is because of the fact that we have, at least part of this is because we have these regulatory T cells. So these T cells can come along and bind onto an antigen-presenting cell that's presenting MHC plus self-peptide, and when these T cells, these regulatory T cells get activated, they then send signals to another T cell that might bind on that antigen-presenting cell that are what we would call conventional cells. They would normally respond and cause an immune response. And through these kinds of suppressive signals then can shut down a T cell response against a particular self-antigen. And we know that these are very important for maintaining what we would call peripheral tolerance, this idea that you need to maintain self-tolerance, not just in the thymus, but also in the periphery. We know that they're important because if you lose genes that are required for the generation of these Tregs, and in particular there's a certain transcription factor that's very important for these T cells to generate and develop into these Tregs. If you lose those guys, they can then go, mice and also in humans develop these multi-organ autoimmune diseases. So, and in humans, we call that IPEX. Now, what does that have to do with the thymus? Well, it turns out that these Tregs, this population of specialized cells, somehow makes its way, most of the cells that are overtly autoreactive are going to get deleted through negative selection. But it turns out that these regulatory T cells are overtly autoreactive. Their T cell receptor binds avidly to peptide and self-MHC, and instead of dying off for some reason that we don't know, we don't understand, instead they differentiate into these regulatory T cells and then maintain that regulatory phenotype for their life. Okay, so that's a really important subpopulation of T cells. We'll come back to that later when we talk about autoimmune disease, but that's something to note is that this process of tolerance that's induced in the thymus is not just about the generation or this negative selection process. It's also about the generation of these regulatory T cells. So just a quick note, I will not be here on Friday. Dr. Fruman will come back for his sort of final appearance in the class, at least until I shouldn't say that. He'll have a final appearance at the final, but he will, that'll be his last lecture. And then I'll come back on Monday and we'll talk about T cell function.